So without any further ado, I'm going to introduce our first speaker, which is Sadra Sadeh. He's based on Imperial College London, and he's going to talk about the theory of neuronal perturbome linking connectivity to coding via perturbations. Sadra, the stage is yours.
Thank you, Elenor, but I'm not capable of sharing the screen yet. Oh, now I can. Perfect, thanks. Cool. Yeah, thank you. In this work, we try to develop a theory of how neuronal perturbations can link to connectivity and coding in cortical networks. As we know, neuronal perturbations are becoming very important and pivotal in our understanding of the brain function. And recently, our perturbation techniques are becoming more complex and more specific. For instance, in this recent study by Chetty and Harvey, they managed to stimulate single neurons in vivo in the primary visual cortex of mouse and measure the impact of such perturbations on the rest of the network. They found that the main influence of neurons on each other is suppressive. Excitatory neurons mainly suppress each other. And this suppression was in fact more for neurons with similar functional properties. For example, neurons who are preferring respond to similar preferred orientations, they respond each other more as a result of single neuron perturbations. It seems to be a bit at odds with recent connectivity data in mouse visual cortex, where we find that excitatory neurons with similar functional properties are in fact connected to each other more and more strongly. To make sense of this conundrum and to come up with a model and theory, we developed a large-scale model of cortical networks in the visual cortex with realistic receptive fields and realistic connectivity profiles as measured in experiments. We stimulated such networks in response to external stimuli in the baseline state before perturbation and after perturbing single neurons in the network. Their change in the response of other neurons in the network as a result of single neuron perturbations was then normalized by the perturbation to obtain a measure of influence. When we plotted that influence as a function of signal correlation or response similarity of neurons in the network, we observed two regimes of interactions. One regime in the intermediate range, where we observed similar feature-specific suppression as reported in the experiments, but there was also another regime of amplification for highly similar neurons. And that regime was in fact also consistent with what the experimentalists found in single neuron perturbations. We could parameterize our networks in terms of different connectivity profiles between excitatory and inhibitory neurons, and by doing a massive parameter search of the parameter space, we could observe that the main ingredient of feature-specific suppression in such networks is the presence of strong and specific E2I connectivity in these networks. We could further develop a theory of how specific motifs of E2I interactions lead to the emergence of such feature-specific interactions and suppressions. And it was very important to understand how connectome is different from perturbome in these networks, especially in networks with a strong interaction of excitation and inhibition. The theory would work for small models and simple models, but also for large-scale recurrent networks of excitatory and inhibitory neurons, where you need to consider all the higher order motifs of interaction between excitatory and inhibitory neurons. I'm happy to expand on this in the discussion if there was any further question. Moreover, we observed that the same motif of connectivity was very important for sensory processing and functional properties of the networks. We presented networks with natural images and look at how networks can distinguish between natural images. And we observed that networks with a strong E2I connectivity and a specific E2I connectivity had the more capacity to decode and encode the natural images. And that was accompanied by the emergence of nonlinearities at the population level, which was important for orthogonalization of responses and population representations. Finally, the theory and model could provide further predictions to be tested in future experiments. These are not tested yet because of technological challenges attached with them. First of all, we could shed light on the temporal evolution of single neuron influence whereby we found that feature-specific suppressions actually emerge later in time, whereas amplifications are present at the beginning. We could also see that in double cell perturbations, when you perturb two neurons at the same time and measure the influence, you see even more suppression, which can be tested further. The theory can also be expanded to more complex situations like multiple cell or pattern perturbations, which would help us in future to map the perturbome of neuronal networks in future experiments. All of this is published now, and if you want to see further details, you can refer to the published paper, but I'm happy to answer any question that you have. In summary, model and theory, we developed a model and theory to study the effect of perturbations in cortical networks and link them to connectivity and coding. And we found that a specific EI connectivity was necessary for feature-specific suppression as measured in experiments. And we could provide further predictions and further expansions of the model and theory that could pave the way to map the perturbation of neurons in future experiments. Thank you.
Thank you very much, Sadra. We already have a question from Marcel. Marcel, if you want, you can unmute yourself and ask a question to the speaker directly, or I can relay it for you as well. So the question is, how do you relate the idea of assemblies being important functional units in the brain and the findings that suppression is the dominant mode between excitatory neurons?
I think that's in the two different regimes that I described. So you have this like dominant regime for most of the neurons, where you have the main effect is suppression, as you see here. So this like consists like a bulk of the population, like 90-something%. But then you have also some very highly like connected assemblies. So you can have different regimes like suppression and amplification based on the degree of like if you wish assembliness. Like most of the neurons are suppressing each other, but then if there is a clique of neurons who are like highly connected to each other, they can amplify each other and stand out of the crowd. But by amplifying each other and also suppressing the rest of the network at the same time. But I think another key point to answer this question is that you should look at the interaction of excitation and inhibition. If you stick to the like a traditional understanding of assemblies where you have only excitation, you cannot understand the suppression. So the most important point here is that assemblies exist not only in excitatory subnetwork, but also in the interaction of excitation and inhibition. And that's very key, because before we were not capable of measuring that excitatory-inhibitory interaction. And that's very important to pinpoint the regimes of interaction.
Thank you very much.
Thank you very much. Yeah.
Oh yeah, Marcel, did you want to ask a follow-up on that? Sorry, I just figured out how to unmute you.
Yeah, so what I wanted to ask was, what do you think, which role do inhibitory assemblies play in that? So assemblies between inhibitory neurons.
I think the first point is that it is very important to have like a similarly matched at least some level of a specificity in the inhibition and their connectivity, because if you form assemblies, they can easily go like crazy. So they can like excite each other and they dominate the network, or they go to like runaway activity. So inhibition seems to be a very good way of like the controlling system naturally. And secondly, it is important to suppress the noise, because if you just amplify the signal within the excitatory neurons, they are not isolated. Like the traditional models, they put them like just binary like subgroups, but in reality, they are connected to other subgroups as well. So when they start to activate each other, they also amplify like a non-relevant subgroups, and they start to become activated, which increases the noise. In fact, that's what we saw in the simulation that I showed you in response to natural images. So if you just put like a weak E to I, like you have a strong E to E connection, but weak to E to I, that's actually worse than feed forward. And that's because you are amplifying the noise. But when you add the strong E to E plus a strong E to I, you not only amplify yourself to a certain extent, but also like suppressing the non-relevant information. So to sum, so first you need them for stability, and secondly, you need them for increasing signal to noise ratio.
Okay, thank you. Thanks.
You're welcome. Thank you for this.
Thank you, Sadra and Marcel. We have two other questions. Oh, 3 other questions we're getting here. I'm going to go by order of appearance. Nicola, you've had a question. Did you want to ask that in person? And I think you're able to unmute yourself.
Yeah, sure. I just wanted to ask whether you do have an intuition why the amplification kicks in earlier than the suppression. Why there is this difference?
Yeah, we analyzed this thoroughly in the paper. Amplification emerges mainly in direct connections, whereas inhibition mainly emerges via polysynaptic interactions, and that's why it is late. So it is actually a hint. I didn't have time to go into that, but you can analyze the motifs of interaction and how amplification and inhibition emerges when you project them over the motifs, and you see that mainly the direct connections are amplification because it is the typical thing that you would expect. But then you have many multiple pathways of higher order motifs that can be either excitatory or inhibitory or disinhibitory, and we can analyze it in the network. But this prediction that they should be late rising, if it is correct, talks to the polysynaptic nature of suppression in the network.
Yeah, perfect. Thank you.
Thank you very much. Sadra, feel free, by the way, later to post a link to your paper in the chat as well. The next question, that was Daniela. No, not Daniela, sorry, Arianna. Arianna, do you want to pose your question yourself?
Hi, yeah, sure. Okay, sorry for the super unknowledgeable question, but it's the first time I've even heard many of these terms. And what I didn't fully get is the perturbome. If we would want to kind of quantify something like that in an actual in a brain, would it be defined kind of on a large network level? So, for example, would we be interested in changes in activity in say a whole functional area upon changing activity of one neuron, or would it be on a small circuit level? I didn't fully grasp that concept.
Well, it is in large scale networks. But typically what we do is we perturb a large scale number of neurons as well because of our techniques. Or because if you stimulate one neuron, it is very difficult to measure the impact of that neuron. Because the network is built of billions of neurons. It's very difficult to stimulate one neuron and effect. And that the whole beauty of the experimental result that they presented was that they could assay some sort of the effect of that they could get some signal out of that noise. But it is also interesting that because it is showing us something that would have been more difficult to get with more complex pattern of perturbations. Mm-hmm. Because when you have more complex pattern of perturbation, you get more interactions, which makes it more difficult to tease out the specific interaction that you get from a single cell. So if we can get that single cell, that resolution and that specificity, we can tell something about connectivity. The perturbome is not exactly like connectivity because of all these higher order interaction, especially when you have excitation and inhibition. But it can, if you have theory and computational modeling, it can bridge the gap between connectivity and function, essentially.
Because you could then extract that from the perturb. Okay, yeah, that makes a lot of sense. Thank you.
Thank you.
Awesome. Next up is Bastian.
Thanks a lot, Sadra, for the talk. I really liked the work.
Thank you.
So I was interested in, do you see any distance dependency of suppression and amplification? Because I'm aware of a lot of work, especially in visual cortex, where they assume for modeling these Mexican head-shaped profiles or this sort of stuff. So do you see something like that, or do you have anything like that in the model at all?
But the Mexican head can be assumed in two spaces. One is the physical space, which is the distance dependence of connectivity. For example, there's a paper similar to this from Allen Institute where they had this actual connectivity profile, and there they could see that distance dependence. And actually, the distance dependence is also described in the original publication, in Chetty and Harvey. So there is some distance dependence. But you can also assume a Mexican in the functional space because of that specific connectivity that I told you. So neurons with similar functional properties, independent of whether they would develop some sort of connectivity to each other. And I think that's more important for us. And that's exactly the difference of a specificity that you allow for excitatory and inhibitory neurons. And if you allow for, and this is something that we didn't present here, but we quantify that, we parameterize that in terms of the specificity of excitatory and inhibitory neurons. And you can see that it is important to have both a specificity of excitation and inhibition, but also you can play with having more specificity in excitation than inhibition, and then you get some sort of sharpening. But it is important to have some degree of specificity. So this is the slide that I was referring to. So you can play with the inhibition dominance in the network, but also with the amount of specificity of inhibition in the network. And there is a regime where you get that like feature-specific suppression. If you have only broad inhibition, you don't get it. You need some degree of specificity of inhibition to get it, but it shouldn't be also too much, because if it is too much, it kills the amplification. Sorry, I didn't have time to talk about this, but this is like thoroughly discussed in the paper. If you wanted to see more details on it, you can refer to it. Okay, we have a little bit more time for a very brief question. Matt, I have allowed you to talk. You should be able to unmute yourself. Sadra, please be fairly quick with your answer here. Sure, sorry.
I'm just interested in the connectivity of how the connectivity relates to the correlation and therefore the amplification or suppression. And I just wondered, would you predict, or do you know whether the highly correlated neurons that amplify share strong sensory tuning, or are they actually sort of more stimulus invariant and might convey other information about a stimulus, for example, or not care about sensory information?
Well, we know that we don't know it from experiments, because I think the reason is that they didn't massively target all the neurons in a smaller scale to be able to tell that. But we know that the noise correlation is more between neurons who are like amplifying each other, which might be going back to the question that maybe the direct connections are actually the amplification, directing the amplification, whereas the inhibition is emerging via polysynaptic synapses. I'm not sure if I answered the question, but.
No, this looks great. Thanks. I'm just, it's a really cool concept, and I'm excited to see how we can sort of expand it to apply to like other sort of maybe more complex connectivity rules that we know of in cortex. And so it's really cool. Thanks.
Thank you guys. Moving on. Thank you so much, Sadra, one more time for this really good discussion and talk as well. Next up is Nicola.